Today, we are diving deep into the structure and function of human skin, which, I have to say, after going through these sources, is without a doubt the body's most massive and yet most systematically underestimated organ.
It's the ultimate paradox, isn't it? We see it every single day, we live in it, but we so rarely appreciate what it actually is.
Which is what?
It's a full-fledged multi-system interface. It's our living armor. And the sources we have here, they're basically a comprehensive medical blueprint. They really demand that we move past thinking of skin as just a covering.
Right, just a wrapper.
Yeah, and start treating it like the critical, incredibly dynamic system it is.
I completely agree. And before we even get into what it does, I think we need to establish the sheer scale we're dealing with, because it's just immense.
It really is.
We talk about the brain or the liver by weight, but get this. If you, our listener, are a 70-kilogram adult, your skin makes up about 15 to 20% of your total body weight.
And just to put a number on that, you're carrying around 10.5 to 14 kilograms of this stuff.
That's the weight of a toddler.
It's the weight of a toddler or a fully loaded hiking pack. It's a serious amount of material. And if you could, lay it out flat, the total surface area is somewhere between 1.5 and 2 square meters.
2 square meters.
That scale alone should tell you something about the magnitude of the work it's performing day in and day out.
And what's so astonishing about that 2 square meters is how incredibly variable it is. We are not dealing with a uniform sheet of material here.
Not at all. The thickness of this living armor changes completely based on the job requirements of that specific body part.
So, for example?
Well, on your eyelids, where you need maximum flexibility and light, quick movement, it can be unbelievably thin. We're talking around 0.05 millimeters.
Wow, paper thin.
Thinner than paper. But then you contrast that with the tough, load-bearing, friction-resistant skin on the soles of your feet or your palms.
Right.
There, it can be up to 4 millimeters thick.
So that's almost 100 times thicker.
An incredible range.
Yeah.
And that extreme variability is the very first clue that the skin is highly engineered for specific local demands. It's not just a single barrier.
It's a mosaic. A mosaic of specialized tissues designed for totally different levels of protection and sensitivity. So I think our mission for this deep dive is clear. We need to move way beyond simple anatomy.
Exactly. We're not just here to list the layers.
No, we're here to figure out the complex, integrated roles of the entire integumentary system.
The skin itself, plus all its appendages, the hair, the nails, all those complex little glands. Our focus has to be on how this system functions as a dynamic regulator, a metabolic engine, and an immune fortress all at the same time.
A constant, coordinated effort.
That's it.
Okay, let's unpack that functionality. We've set the stage with the scale, but what are the essential jobs that really elevate the skin into the category of a major organ system?
Well, it's the ultimate multitasking powerhouse. And what's so fascinating is how it manages dozens of crucial, simultaneous roles without ever really failing.
So to make sense of it, you suggested we can break it down.
Yeah, we can break its massive job description into 3 core categories: protective, regulatory, and metabolic. And protection is, where it all begins.
Which makes perfect sense. It's the front line. It's the part of us that's in constant direct contact with what is often a pretty hostile environment.
Precisely. So first and foremost, there's the physical barrier. The outermost layer, the part we see and touch, is incredibly tough. It provides really effective mechanical resistance against trauma.
So cuts, scrapes, impacts.
All of it. This is our primary defense that stops that kind of damage from reaching our deeper, more vital organs.
Now, I found the statistic on fluid balance in the sources to be absolutely mind-boggling. We just don't think about our skin as this thing that regulates how much we dry out.
But it is probably its most vital, most immediate function. The skin acts as a highly selective, basically waterproof barrier.
Okay.
If you didn't have this intact barrier, you would lose water from your body at a completely unsustainable rate. The skin prevents approximately 90% of insensible fluid loss.
90%. So what does that mean in practical terms?
It means that if you were to, say, remove the skin, you would desiccate. You would dry out almost instantly. So you really have to think of the skin as a microscopic, continent-spanning dam holding back this vast internal reservoir of water that's absolutely necessary for life.
And the regulatory part of that barrier means it's not just holding water in, it's deciding what gets in from the outside.
Yes, it tightly regulates absorption. And this is really a double-edged sword. On one hand, it allows medical scientists to design things like transdermal patches to deliver certain drugs through the skin effectively.
Right, like nicotine patches.
Exactly.
Hmm.
But at the same time, it ensures that most environmental toxins and harmful chemicals can't just breach the barrier and waltz into our bloodstream. It's a very selective, very intelligent bouncer at the club door.
Okay, so let's talk about UV protection. This is probably the defense that we're most actively conscious of, especially in the summer. How does the skin handle that constant invisible threat?
This is one of its most sophisticated and adaptable defenses. It uses specialized cells, the melanocytes, to produce melanin,
The pigment.
the pigment. And this melanin, it's a complex polymer. It does two essential things. First, it physically absorbs UV radiation, dissipating that energy as harmless heat before it can penetrate and damage the DNA in our cells.
Second.
Second, it acts as a radical scavenger. It helps to neutralize the reactive free radicals that are generated by sun exposure, which further minimizes DNA damage. It's an incredibly effective, built-in solar shield.
So we have physical resistance, we have waterproofing, and the UV shield. But the sources also make it really clear that the skin isn't just a passive wall. It's an active part of our immune system.
It's a crucial primary immune organ. It can initiate both innate and adaptive immune responses right there at the surface.
Okay, so what's the innate part, the always-on defense?
In terms of innate immunity, it's always ready. It has its own resident immune cells, including specialized Langerhans cells and T cells that are positioned like sentinels within its layers, constantly surveying for threats.
But I found the biochemical defense is fascinating, the idea that it produces its own natural antibiotic.
It does. Beyond the cellular sentinels, the skin actively secretes this powerful cocktail of defense molecules known as antimicrobial peptides.
What are those?
The sources specifically mention defensins and calcitins. These are small proteins that actively punch holes in the cell walls of potential invaders. They're known, for instance, to protect us against very common and potentially serious pathogens like Staphylococcus aureus, and they also inhibit various types of fungal growth.
So that's the first line of defense. But if that physical barrier is breached and a serious threat gets through, the skin is what initiates the systemic warning signal, the full-scale inflammatory response.
It absolutely is. The skin is fully capable of initiating and then tightly regulating inflammation. This localized response involves stimulating angiogenesis. That's the creation of new blood vessels needed to bring repair cells and nutrients to the site of injury.
And it's sending out chemical signals.
A whole host of them. It releases signaling chemicals that act as a command center, telling inflammatory cells to stay on site to keep fighting. It affects how blood vessels dilate or constrict, immediately recruiting systemic forces from all over the body to the breach.
Okay, so moving from protection to the regulatory roles. Thermoregulation is something we feel every single day. When we're hot, when we're cold, the skin is in charge.
Thermoregulation is arguably its most critical regulatory function for our survival in changing temperatures. The skin accounts for a staggering 90% of the body's total heat loss.
90%.
The entire process hinges on controlling blood flow to the surface, and it does this with extreme precision and incredible speed.
Okay, so when I'm freezing cold, my fingers are turning white. How aggressively is the skin actually cutting off that blood flow to conserve my core temperature?
It's incredibly dramatic. In a cold response, the blood flow to the skin can decrease by as much as 20 times the resting rate.
20 times.
It's a life-saving mechanism. It essentially clamps down on all those tiny blood vessels, diverting the warm blood deep into the core organs, the torso and head, basically sacrificing the comfort of the periphery, like your fingers and toes, to ensure the survival of the core.
And what about the opposite? When we're exercising or it's a hot day and we're overheating, how does the skin transition into being this massive emergency radiator?
When you overheat, those same vessels open up dramatically. Blood flow increases significantly, bringing all that core heat right to the surface where it can be dissipated into the environment through convection and, more importantly, through the evaporative cooling from sweat.
And how much of our blood supply can it actually demand?
During peak heat stress, the skin's blood vessels can demand and receive up to 5% of the total cardiac output.
5% of all the blood pumped by the heart.
Yes, just to dump heat at the surface.
Yeah.
That is a massive volume of blood dedicated solely to temperature control.
That is genuinely astonishing. We think of the skin as so passive, but it's capable of redirecting a major portion of our entire circulatory power.
It's anything but passive.
Yep.
And finally, we have to touch on the metabolic and endocrine roles. The most famous of these, of course, is vitamin D synthesis.
Right. I think most people know the skin is involved, but they might not realize that it is the single most essential starting point for the whole process.
It is the foundational chemical factory for vitamin D. The skin is the only site in the entire body where UVD exposure from the sun can act on a precursor molecule called 7-dehydrocholesterol,
Okay.
and it converts it into vitamin D3. This initial transformation must happen in the skin. From there, the vitamin D3 travels to the liver and the kidneys for final processing into the active hormone. But without that initial cutaneous step, the whole chain reaction just stops.
So the skin is literally manufacturing a vital hormone for us. And the sources hint at other metabolic involvement too.
They do. It's involved in broader lipid and hormone metabolism, including the processing of androgens, which are crucial for sexual development, and our patterns of hair distribution.
And of course, we can't forget its role as our major sensory organ.
Absolutely not. It is supplied by this incredibly dense and complex network of nerves and specialized receptors.
And that system is what allows us to communicate sensation, pain, temperature, touch, vibration, pressure,
Right.
which is essentially how we interact safely with the world around us.
Exactly. The skin provides the complex feedback loop that is necessary for skilled manipulation of objects, for avoiding injury, and just for maintaining basic situational awareness. It is constantly monitoring our internal state and the external environment all at the same time.
Okay, so here is where we go microscopic. We're going to delve into the epidermis now, that outer layer. The surface we actually see is just the very tip of this highly structured, constantly self-renewing stack. It is a remarkable piece of biological engineering.
It really is.
So let's start with the workhorse cell you mentioned earlier, the keratinocyte.
The epidermis is a masterpiece of organized, programmed self-destruction. It's what's called a stratified epithelium, meaning it's arranged in very distinct layers. And the sole purpose of the keratinocyte is to be born at the very bottom, to mature as it moves upward, accumulate protective material, and then die to form that tough outer barrier.
And what's the typical timeline for that journey, from its birth to its death and sloughing off?
It's typically about 28 days. It's a highly precise, continuous 4-week migration from the bottom to the top. They are constantly pushing upward. And this process, which is called keratinocyte turnover, is intensely regulated.
And its disruption tells us a lot about skin health.
A huge amount.
Wow.
When that timeline is dramatically accelerated, for example, we see rapid pathology.
Such as?
In chronic inflammatory conditions like psoriasis, that turnover time can be drastically shortened, sometimes to just a few days instead of 28. The cells are produced and pushed up so quickly that they just don't have time to properly mature. And that leads to this rapid, disorganized buildup of uncompacted, flaky scales that characterize the disease. It's like a factory floor working at triple speed, but with zero quality control.
So let's start that journey at the very bottom, at the deepest layer, the stratum-based cell. This is the foundation and the factory floor all in one.
This is the home of proliferation. It's a single layer of cells that contains the keratinocyte stem cells. These are the cells that are continuously dividing to replenish all the layers above them. They are columnar in shape, and they exhibit intense mitotic activity.
Cell division, basically.
The constant cell division that's necessary to fuel the entire 28-day process.
And these cells, they need to be absolutely bolted down to the foundation, right? Otherwise, when the skin moves or stretches, they just float away.
They have to be anchored with extreme structural integrity. They use these specialized adhesion complexes called hemidesmosomes, you can think of them as molecular rivets, to attach very strongly to the underlying foundation.
And that foundation is called the dermal-epidermal junction.
Yes, or the basement membrane zone, the BMZ.
So what's the consequence if those rivets fail?
Failure here is disastrous. If those attachments, which involve complex molecules like laminin and collagen S70, if they're compromised by genetics or trauma, the epidermis will instantly shear off the dermis.
And that's a blister.
That is the physiological basis of blistering diseases. It's also why our skin is so sensitive to friction injury, especially when it's wet. The integrity of that basal anchor is absolutely non-negotiable for skin health.
Okay, so once they divide, they get pushed up one layer to the prickle layer, or the stratum spinosum, and this is where they start developing their characteristic appearance.
Indeed. As the cells migrate upwards, they start forming massive numbers of intercellular connections called desmosomes.
Desmosomes.
These are incredibly powerful spot wells that form between adjacent cells. Now, when skin tissue is prepared for a microscope slide, the cells shrink a little bit, but they hold onto each other fiercely at these desmosomal junctions.
And that's what gives them that spiny look.
That's exactly what gives them that classic prickle cell or spine-covered appearance. This network of connections provides huge mechanical strength to the tissue, allowing the skin to withstand pulling and tension without tearing apart.
And inside the cell, the scaffolding is already changing to prepare it for its future as a tough outer shield.
Yes, this is the first major step in differentiation. The cells here begin to shift their internal keratin scaffolding. They replace the building keratins used in the basal layer with the toughness keratins, the suprabasal keratins. This marks the true start of their final maturation into the protective shield.
And we also see some other cells in this layer.
Yes, this is where we find our resident immune cells, the Langerhans cells, and T cells, scattered throughout, performing constant immune surveillance.
Okay, next up, we hit the granular layer, the stratum granulosum. Now, this is described as the crucial transition layer. It's usually only 3 or 4 cell layers thick, but everything essential to waterproofing happens right here.
This is the chemical factory. This is where the final barrier is assembled. At this point, the keratinocytes stop dividing, and they begin synthesizing and packaging these specialized granules that are going to define the skin's function. And there are 2 major types.
The first type, I guess, builds the interior of the brick in the brick and mortar model.
Right. These are the keratohyalin granules. They contain a very important protein called profilagrin. As the cell matures and moves up, profilagrin is processed into its active crucial form, filigrin.
And what does filigrin do?
Filigrin's job is to aggregate all the individual keratin filaments into these dense, tightly packed bundles that are required for the tough outer layer. So it's the internal mortar defining the structural integrity of the brick itself.
And the second type of granule creates the external cement, the stuff that actually seals the whole surface.
Exactly. These are the lamellar granules, sometimes called odd-land bodies. These organelles move to the cell surface and release a very precise mixture of complex lipid ceramides, phospholipids, glucosylceramides into that narrow space between the cells.
And that's the waterproofing step.
This is the ultimate waterproofing step. This secreted material is rapidly processed into this continuous essential lipid matrix that fills all the gaps between the cells. This process is what transforms the skin from just a structural sheet into a truly impermeable barrier, drastically limiting water loss.
And this link between the granular layer and clinical conditions is incredibly important for anyone who deals with dry skin or eczema.
It is the ultimate so what moment of this whole process. We now know that defects in the gene that codes for filigrin, the FLG, result in a compromised leaky barrier.
So if you don't make good filigrin,
If you don't make good filigrin, the bricks are structurally weak and the overall barrier is compromised from the inside out. This genetic vulnerability is known to significantly contribute to common dry skin conditions like ichthyosis vulgaris, which is characterized by scaly dry skin.
and it also plays a role in eczema.
It plays a major predisposing role in atopic dermatitis or eczema.
So eczema isn't just a surface irritation then. It's often a failure of the internal construction machinery, which makes the skin leaky and allows environmental allergens to constantly penetrate and trigger that inflammation.
That is the precise mechanism. The compromised barrier means the skin is vulnerable to chronic water loss from the inside and constant infiltration by irritants from the outside, leading to the redness, itching, and scaling that we associate with the condition. It just highlights how incredibly important that tiny 3 to 4 cell thick layer is for overall health.
And finally, we reach the surface, the cornified layer, the stratum corneum. This is the protective shield we see and touch every day. Structurally, this is the end result of that whole 28-day journey.
This is the classic brick and mortar model realized.
Yeah.
The bricks are the dead, flattened, fully keratinized cells called corneocytes.
Okay.
And they are just packed with those dense keratin filaments that were aggregated by filigrin. And the mortar is that continuous, impermeable lipid matrix that was secreted from the granular layer.
And the thickness here is highly responsive to friction and use.
Absolutely. In thin skin, like on your forearm,
Right.
you might have 10 to 15 cell layers, which is pliable and delicate. But on the palms and soles, where friction is constant, this layer can be up to 50 cell layers thick,
Wow.
giving you that tremendous toughness and resilience required for gripping things and for walking.
So how does this layer, which is made of dead cells, maintain its flexibility and avoid just cracking like dry mud?
It's a great question, and it relies heavily on the internal components of those dead corneocytes. The breakdown products of filigrin, which we just discussed, actually serve another purpose. They become crucial natural moisturizing factors, or NMFs.
Moisturizing faster.
Yes, and these NMFs are highly hygroscopic. They literally pull water from the air and from the underlying layers of the skin, trapping it inside the corneocytes. And it's this internal hydration that maintains the necessary flexibility, preventing that tough outer layer from becoming brittle and fracturing.
Beyond that sheer mass of keratinocytes, the epidermis hosts a few specialized residents that handle the more advanced, non-structural tasks, producing color, providing immune defense, and sensing touch. Let's look at the roles of these specialized operators.
Yeah, these are strategically placed cells that operate at the intersections of protection, communication, and our response to the environment.
Let's start with the melanocytes, the pigment engineers. Where are they located, and how do they physically deploy that UV shield we talked about?
Melanocytes are the deepest of these specialized residents. They're mainly located in the basal layer, nestled right among those basal keratinocytes, and they're dendritic cells, which means they look a bit like a spider with these long branching arms that reach out to surround about 30 to 40 of the neighboring keratinocytes.
And they manufacture the shield inside these specialized chemical packages.
Correct. They synthesize that complex melanin polymer inside their own internal factories, which are called melanosomes. Once a melanosome is mature and filled with pigment, the melanocyte transfers it directly into the body of the surrounding keratinocyte.
How?
Through a process called cytokine secretion, it literally injects the melanosome into the other cell.
So the keratinocyte acts as the ultimate delivery device and the holder for the shield itself.
That's the perfect description. And once inside the keratinocyte, the melanosomes don't just scatter around randomly. They cluster together, and they position themselves directly over the cell's nucleus
Ah, to protect the DNA.
to form a dense, protective cap known as the supranuclear cap. This acts like a microscopic parasol, perfectly shielding the cell's precious DNA from harmful UV radiation, which is the entire point of the system.
And this brings us to the vast spectrum of human skin color. If I have light skin, does that mean I just have fewer melanocytes than someone with dark skin?
This is a really common misconception, and it's so important to clarify. The total number of melanocytes is roughly constant across all racial groups and all skin types.
Okay, so what creates the difference?
The dramatic variations in color, from very fair to very dark, are primarily due to three factors: the amount of melanin that's produced, the size and distribution of the melanosomes themselves, and crucially, the type of melanin being produced.
So it's about the efficiency and the specific chemistry of the pigment, not the number of factories.
Exactly. Darker skin tends to produce more of the dark brown-black humelanin, which is highly efficient at absorbing UV light. Their melanosomes are typically larger, more intensely pigmented, and they persist much higher up into the spinous layer of the epidermis.
And lighter skin.
Lighter skin types tend to produce more of the lighter red-yellow pheomelanin. Their melanosomes are smaller, less pigmented, and they often get broken down and degraded much more quickly after being transferred to the keratinocytes.
And what about the immediate tanning response versus the delayed response we see after being in the sun?
So when you first step out into the sun, the immediate tanning you might notice is caused by a quick physical redistribution and photooxidation of the melanin that's already there. That's why you might look slightly tanned after just an hour. But the delayed tanning response, the one that becomes really noticeable after a few days, that's the significant, long-lasting change. This involves an actual increase in the synthesis of new melanin and an accelerated transfer process. You're essentially expanding the protective shield in response to a sustained UV insult.
Okay, now let's shift to the Langerhans cells. These are the full-time immune sentinels. Where are they posted, and what is their mission?
They are the ultimate scouts. They patrol the mid-region of the epidermis, so they're primarily located in the stratum spinosum, the prickle layer. And they are bone marrow-derived, highly mobile dendritic cells. They make up about 2 to 3% of all epidermal cells.
And their function is surveillance, but they also have a key role in communicating threats to the rest of the body, right?
That is the crucial part. They function as highly effective antigen-presenting cells, or APCs. They are constantly sampling their environment, and they capture invading antigens from a virus, a bacteria, or even simple environmental allergens, like the chemicals in poison ivy. And once they've captured the threat, they rapidly exit the epidermis, migrate down into the dermis, and then they travel via lymphatic vessels to the local lymph nodes.
And that's where the handoff happens.
Yes. Inside the lymph node, they present the captured antigen to T lymphocytes, and that's what initiates the highly specific, large-scale adaptive immune response. They are the absolutely indispensable bridge between the skin's local defense and the body's entire systemic immunological memory.
Which is what gives us long-term immunity to things.
Exactly.
And finally, in this group, we have the Merkel cells. These aren't involved in defense or color, but they are critical for our sense of touch.
Merkel cells are our specialized touch detectors. They're found down in the basal layer, and they often concentrate in highly sensitive areas like our fingertips and around hair follicles, places where fine tactile discrimination is essential.
And they're always found with a nerve ending, right?
Always. They're found in very close association with specialized nerve endings.
And together, what kind of sensation do they provide?
The Merkel cell and its nerve ending form a functional unit that acts as a slow-adapting mechanoreceptor.
What does slow adapting mean?
It's distinct from, say, feeling a vibration, which is fast. Slow adapting means they excel at registering sustained touch and pressure. They keep firing as long as the pressure is there. They are the reason you can accurately feel the texture of a piece of fabric or maintain a precise, steady grip on a delicate object without constantly losing and regaining that sensory input. Okay, so if the epidermis is that self-renewing, specialized armor plating, the highly organized surface, then the dermis is the underlying high-tech scaffolding. It's the engineering support system and the communication hub for the whole operation.
Right. This is where the true tensile strength, the elasticity, and the deep connectivity of the skin really live.
Exactly.
So what exactly provides that incredible strength and recoil? It's not the cells themselves, but the matrix they produce.
Correct. The dermis is almost entirely defined by its extensive, complex extracellular matrix, or ECM. Its incredible tensile strength comes mainly from collagen.
Which type?
Specifically type I collagen, which makes the vast majority of its protein. You can think of collagen fibers as these braided steel cables that provide immense resistance against tearing forces.
But of course, the skin is also flexible. It stretches and then it snaps right back.
And that's the function of elastin fibers. They are integrated throughout that collagen network, and they provide the essential recoil capacity that allows the skin to be deformed by smiling, by bending a knee, by gripping something, and then return immediately to its original shape.
And then there's this gel that holds it all together, the ground substance.
Yes, the ground substance is a viscous gel that binds everything together and fills all the space.
What is its main function?
It's composed of these molecules called glycosaminoglycans and proteoglycans. And their primary role is to hold vast amounts of water. This water retention is what provides the skin's volume, its resilience, and its essential turgor, its firmness. When skin gets dehydrated, it loses this volume and starts to wrinkle much more easily.
And the workers that maintain this entire massive scaffolding system.
Those are the fibroblasts. They're the primary cellular component. They're the factory workers that are constantly synthesizing and remodeling all those matrix components, the collagen, the elastin, and the ground substance. The dermis also hosts a significant population of resident immune cells, like macrophages and mast cells, ready to initiate a defense deep beneath the surface.
Structurally, the dermis is organized into two distinct layers, the papillary and the reticular. Let's start with the superficial one, the papillary layer.
The papillary layer is the thin, loose connective tissue that lies immediately beneath the epidermis. And this is where that critical mechanical locking occurs.
Right. It's not a smooth junction, which seems critical for preventing the layers from just sliding apart.
It is absolutely crucial for that. The dermis pushes upward into these ridges called dermal papillae, while the epidermis extends downward with epidermal ridges.
Like an egg carton.
It's often compared to the interlocking, finger-like projections of an egg carton, and this pattern massively increases the surface area for nutrient exchange between the layers. But more importantly, it mechanically locks the two layers together.
And this makes it resistant to what kind of force?
It makes the skin highly resistant to shearing forces, which are the forces that try to slide the epidermis off the dermis. This is why a simple touch or rubbing doesn't immediately cause a blister.
And what key sensory structures do we find in this superficial layer?
Well, a papillary layer is highly vascularized with tiny capillaries to feed the avascular epidermis above it. And it specifically houses the delicate Meissner's corpuscles, which we'll discuss more later. These are encapsulated receptors that specialize in sensing light touch and low-frequency vibration.
Okay, so moving deeper, we hit the massive load-bearing layer, the reticular layer.
The reticular layer provides the bulk of the skin's volume and its real strength. It's composed of dense, irregular connective tissue. Here, the collagen fibers are much coarser, and they're organized into these thick bundles that run in specific directions, generally parallel to the skin surface.
And that specific fiber organization has real-world surgical implications, leading to what are called tension lines.
That's right. These bundles create what are known as Langer's lines, or cleavage lines. And surgeons are acutely aware of these because an incision that's made parallel to these tension lines will heal with much less scarring and tension than a cut that's made perpendicular to them.
And this layer contains a lot more than just collagen.
Yes, This layer also contains most of the skin appendages, hair follicles, glands, as well as the largest blood vessels and the deep pressure receptors, the Pacinian corpuscles.
Finally, beneath the dermis, we find the hypodermis, or the subcutaneous tissue. Functionally, it's inseparable from the skin, even if technically it's its own layer.
The hypodermis is essentially a layer of loose connective tissue and adipose tissue, what we commonly call body fat, and it serves three main vital functions.
Energy storage, insulation, and shock absorption.
Exactly. It's the primary site of our long-term energy storage, holding triglycerides. It provides vital thermal insulation, preventing excessive heat loss from our core, especially in cold environments. And it acts as a critical mechanical shock absorber, cushioning our underlying bones and muscles against impact.
And the distribution of that energy store is highly influenced by genetics, age, and sex.
It's highly variable. We see significant sexual dimorphism in fat distribution. Females tend to have greater deposits in the thighs and buttocks, while males often concentrate deposits more in the abdominal area. The sheer volume and placement of this subcutaneous fat also dictates skin mobility, how easily the skin can slide and move over the deep fascia underneath.
Okay, so we've covered the layers, the scaffolding, and the anchoring mechanisms. Now, what does this whole system mean for the specialized structures that we actually grow: hair, nails, and glands? These appendages are all downgrowths of the epidermis that originate deep in the dermis or even the hypodermis, but they handle very unique, complex tasks.
These are specialized structures, and they're dedicated to things like lubrication, secondary protection, and very precise thermoregulation. Let's start with the pilosebaceous unit.
Which is the hair follicle and its associated sebaceous gland.
That's right. The two are a functional unit.
The development of the hair follicle sounds like a complex communication process between the epidermal cells and the dermal layer below.
It is a fundamental process of tissue communication. The hair follicle itself is a deep epidermal downgrowth that forms this highly proliferative structure at its base called the hair bulb. And crucially, the hair bulb cups a structure called the dermal papilla.
Why is that dermal papilla so important?
The dermal papilla is the control center. It contains the blood supply and the connective tissue necessary to continuously deliver essential signaling molecules and growth factors to the cells of the hair matrix.
And the hair matrices?
Those are the highly proliferative cells within the bulb that actually build the hair. If you remove the dermal papilla, the hair stops growing instantly. It's what dictates the hair's entire life cycle.
Let's briefly look at the structure of the hair shaft itself. It's a marvel of protein engineering.
It's incredibly tough. It's composed of three concentric zones of highly keratinized cells. You have the inner medulla, which is usually only present in thick hair, then the massive middle cortex, which contains most of the bulk, strength, and the pigment of the hair.
And the outside layer.
And the outermost layer is the cuticle, which is a layer of overlapping scale-like cells that provide protection and give the hair its luster.
Now, for the hair growth cycle, it's highly regulated, and thankfully for us, it's asynchronous.
That asynchrony is key. It means that if you have 100,000 hairs on your head, they're all at slightly different stages in the cycle, so they don't all fall out at once.
And the cycle has three main phases.
Yes. First is anagen, which is the active growth phase. On the scalp, this can last anywhere from 2 to 7 years. During this time, the cells in the hair matrix are undergoing rapid mitosis, pushing the hair out steadily.
And when that long growth phase finally ends,
It transitions into catagen. This is a brief regression phase that lasts only a few weeks. The proliferative activity just stops. The lower portion of the follicle begins to degenerate, and it detaches from that vital dermal papilla. The hair has effectively committed to stopping its growth.
and that leads into telogen, the resting phase.
Telogen can last for several months. The hair, which is now called a club hair, is held passively in the follicle sheath until the next cycle begins. And when the follicle returns to anagen, the new proliferative hair simply pushes the old club hair out.
And this entire cycle is profoundly influenced by hormones.
Particularly androgens, which dictate where we grow thick terminal hair versus fine vellus hair, creating the typical patterns of sexual dimorphism we see between men and women.
Now, attached to most of these hair follicles are the sebaceous glands. These are responsible for the oiliness of our skin.
Sebaceous glands are small alveolar structures, and they're typically connected to the side of the hair follicle to form that pilosebaceous unit. Their defining characteristic is their secretion mechanism, which is called holocrine.
Can you explain holocrine secretion clearly?
It's pretty dramatic. It means the entire secretory cell, the sebaceous matures, fills completely with lipid droplets, and then it completely self-destructs, it lyses, to release its contents into the duct.
So the final product, sebum, is literally the entire dead cell and all of its accumulated lipids.
Exactly.
And sebum then performs three critical functions once it's released onto the skin surface.
Right. It lubricates the hair shaft and the skin surface, keeping them pliable. It helps to maintain the overall integrity of the epidermal barrier, reinforcing that waterproofing. And importantly, it possesses antimicrobial properties, which helps prevent the colonization of the skin by various microorganisms.
And the activity of these glands is strongly regulated by the changes we experience during puberty.
Absolutely. Sebaceous gland activity is minimal during childhood, but it increases dramatically starting in adolescence, and is driven directly by that surge in androgens. This dramatic increase in sebum production, coupled with changes in the duct lining, is the reason why acne is so common during these formative years.
Now, for the most widespread appendages, the sweat glands, we really need to differentiate between the two types here, eccrine and apocrine. Let's start with the eccrine glands, which are our primary thermoregulators.
Crane glands are by far the most numerous. We carry between 2 and 5 million of them distributed all across our body, and the highest concentration is surprisingly found on the palms and soles.
And their ducts open directly onto the skin.
Yes, directly onto the skin surface, totally separate from the hair follicle.
And their entire purpose is just simple, rapid cooling.
Yes, through evaporative cooling. They secrete a watery, initially isotonic fluid, but they are incredibly efficient. As that fluid travels up the duct toward the surface, sodium and chloride are passively reabsorbed back into the body.
So the sweat that comes out is less salty.
Right. It results in a slightly hypotonic or less salty than blood sweat arriving at the surface. And they're innervated by sympathetic nerves that use acetylcholine, which means they are primarily responsive to heat and exercise stimuli.
And then we have the apocrine glands, which are much more localized and serve a different, perhaps more vestigial function.
Apocrine glands are restricted to specific, often hairy areas.
Yeah.
The axilla, so the armpits, the groin, and the areola. And a key difference is that their ducts always open into the upper part of the hair follicle, making them part of that pilosebaceous unit.
And their secretion is different in its composition as well.
It's much more viscous, and it's rich in organic components, proteins and lipids. And crucially, the secretion itself is initially odorless. It only develops a pungent odor when it is broken down and metabolized by the resident bacteria on the skin surface.
And their trigger mechanism is different from the eccrine glands.
Yes. They are innervated by sympathetic nerves that use adrenaline adrenergic fibers, meaning they respond specifically to emotional stimuli and stress. This is why emotional sweating, the cold sweat you get during panic or fear, originates largely from these apocrine glands.
So what's their function?
Well, their exact human function is debated, but they are hypothesized to have vestigial pheromonal roles.
Finally, the nail apparatus, the hard keratinized plates on our fingers and toes.
The nail is a specialized structure that provides protection for the tips of our digits and also aids in fine manipulation and scratching. It's essentially a hard, dense plate composed entirely of tightly packed, fully keratinized epithelial cells.
And where is it formed?
It's formed by the highly active nail matrix, which is located deep beneath the proximal nail fold, or the cuticle area. Its growth rate is continuous, but it's influenced by everything from environmental trauma to age and your circulation. The incredible functional complexity we've been detailing, from thermoregulation to sensation, it all depends entirely on the sophisticated infrastructure running just beneath the epidermis. The skin is only able to act as this ultimate regulator and sensory interface because of the way its blood vessels and nerves communicate so rapidly and precisely with the rest of the body.
Let's look at that vascular supply. That ability to shift massive volumes of blood so rapidly for temperature control really is a feat of engineering.
It truly is. The blood supply to the skin is highly adaptable. We mentioned it can decrease flow by 20 times to conserve heat or dramatically increase it to dissipate heat. And this is possible because the vessels are organized into a highly structured network of plexuses.
Which are arranged horizontally, kind of like underground highways.
Precisely. The blood vessels run horizontally in two primary networks. The main highway is the deep subdermal plexus, which runs at the boundary between the dermis and the hypodermis.
Okay.
Branches rise from this to form the secondary highway, the superficial plexus, which is located at the junction of the papillary and reticular dermis. From this superficial plexus, tiny little capillary loops extend upwards into the dermal papillae, getting incredibly close to the avascular epidermis to provide the nutrients and oxygen necessary for that constant turnover.
And the mechanism for rapid heat dumping or heat conservation involves these specialized shortcuts.
These shortcuts are the essential key to thermoregulation. They are the arteriovenous shunts, or AV shunts. These are specialized connections that allow arterial blood to bypass the capillary loops entirely and flow directly into the small veins.
Where are they most common?
They're most numerous in areas that are critical for rapid heat exchange, like the fingers, the toes, the palms, and the soles.
So they act like emergency bypass valves in a plumbing system?
That is the perfect analogy. When you are cold, these shunts are closed through vasoconstriction. Blood is forced to travel the long way through the capillary loops, but because the vessel openings leading into the skin are also constricted, flow is minimal and heat is conserved internally.
And when you're hot.
When you overheat, the vessel openings dilate and these shunts are opened, allowing massive amounts of blood to flood the surface capillaries to rapidly dump heat. It's the body's emergency thermostat.
And that rapid diversion mechanism allows for the near instantaneous response to thermal stress that we feel constantly.
It ensures that the skin can modulate temperature without placing an unnecessary regulatory burden on the entire systemic circulatory volume. It's a very efficient local system.
Now let's move to innervation. The skin's sensitivity is so complex, it has to distinguish between a feather's touch, deep pressure, and a sharp pain.
The skin is incredibly richly innervated. It's responsible for gathering almost all of our tactile data about the world. And this complex sensitivity relies on several different types of nerve endings, each of which is optimized for a specific type of information.
We have these specialized encapsulated receptors that deal with mechanosensation physical touch. Let's start with the deepest ones.
Deep down in the dermis and the hypodermis, we find the Pacinian corpuscles. These are large, encapsulated receptors that, under a microscope, look a bit like sliced onions.
Okay.
Their physical structure makes them perfectly suited to sense deep pressure and rapid vibration. They are classic fast-adapting receptors.
What does that mean, fast adapting?
It means they respond intensely to the onset and the offset of a stimulus, but they quickly stop firing if the pressure is constant. They tell you that something just happened, not that it's still happening.
Contrast that with the receptors for lighter touch.
Those would be the Meissner's corpuscles. These are smaller encapsulated receptors found specifically up in the dermal papillae, right near the surface. They sense light touch and low-frequency vibration, and they are essential for detailed tactile discrimination, like feeling the fine differences in texture.
And then we have the most numerous and perhaps the most important for warning us of damage, the free nerve endings.
These are the simplest and most pervasive nerve endings. They are unmyelinated or sparsely myelinated, and they are found absolutely everywhere, extending throughout the dermis and sometimes even into the basal epidermis.
And they detect what?
They are the non-specialized detectors, and they are responsible for registering those crucial survival sensations: pain, temperature extremes, and the sensation of itch.
And finally, there's the motor innervation, the automatic systems that run the machinery even when we aren't thinking about it.
That's the autonomic nervous system controlling the smooth muscle. Autonomic fibers supply the smooth muscle of the blood vessels, controlling that vasoconstriction and vasodilation for temperature. They also innervate the tiny erector pili muscles,
The goosebump muscles.
the muscles that contract to make your hair stand on end, causing goosebumps. And of course, they supply the sweat glands, regulating sweat production in response to heat or emotional stress.
This incredible system, it isn't static. It grows, it changes over decades, and most importantly, it repairs itself through a highly dynamic and choreographed process. Let's look at the key milestones of its life, starting with its earliest development.
Skin development is a foundational process that shows how two very different types of tissue can integrate perfectly. The epidermis, the outer layer, originates from the ectoderm, that's the outer layer of the embryo,
Okay.
while the dermis and the hypodermis derive from the mesoderm, the middle layer.
So you have an outer layer coming from one source and the inner foundation coming from a completely different one. That coordination must be intense for appendage development, like for hair.
It's intense tissue signaling. Epidermal-mesenchymal interaction is the key driver for all of it. The formation of complex appendages like hair follicles requires the epidermis to send signals down to the underlying dermis, which causes a specific type of tissue rearrangement, which eventually forms the structures we talked about earlier.
And what about neonatal skin? Is it robust right from birth?
Neonatal skin is significantly immature and quite vulnerable. It's generally thinner than adult skin, and that crucial structural anchoring system, the dermal-epidermal junction, or BMZ, is initially poorly developed.
And that makes it prone to what?
It makes infant skin much more prone to separation and blistering from even minor friction or trauma.
And that thinness must have major clinical implications for things like topical creams and medications.
It does. Because the stratum corneum is less mature and the lipid composition is different, neonatal skin is much more permeable. It absorbs topical substances, including medications and chemicals, much more readily than adult skin does.
And that can be dangerous.
This increased permeability makes infants highly vulnerable to systemic toxicity from products that are applied externally, and also to rapid water loss.
Okay, so moving on to the long game. Aging. Our sources make a very clear distinction between the unavoidable consequences of time, or intrinsic aging, and the avoidable damage caused by the sun, which is called photoaging.
And this distinction is crucial for understanding the visible changes we see in our skin. Intrinsic aging, or chronological aging, is the gradual decline that's purely due to the passage of time. We see an overall thinning of the skin because the density of epidermal cells decreases, and critically, that dermal-epidermal junction flattens out.
What's the consequence of that flattening in daily life?
It makes the skin much more fragile. Remember those interlocking ridges that resist shear forces? As that interface flattens out, the structural integrity weakens, making the skin much more susceptible to tearing, bruising, and injury from simple friction or impact.
And we lose collagen, too.
We see a generalized loss of collagen and elastin and reduced glandular function, which leads to chronic dryness. The skin is literally less securely anchored and less resilient with each passing decade.
And then we have photoaging, which is a complete acceleration and restructuring of the skin due to cumulative UV exposure.
Photoaging is far more devastating structurally than chronological aging is. UV radiation causes disproportionate damage, particularly down in the dermis. This leads to a condition called solar elastosis.
And what is that?
It's where the elastin fibers become clumped, disorganized, and they completely lose their ability to recoil. The skin acquires that classic leathery, wrinkled appearance, coupled with mottled, uneven pigmentation and deep wrinkles. The damage is pervasive, and it accelerates all the other aspects of aging.
So now let's turn to the most dynamic process of all, wound healing. When we suffer a cut, the body initiates this highly choreographed response involving millions of cells and dozens of signaling molecules. What are the 4 phases of this precise repair process?
It's a continuous overlapping cascade that's designed to restore the barrier as quickly and effectively as possible. And it starts immediately with hemostasis.
Phase 1, stopping the bleeding and laying the first provisional track for repair.
Within minutes, platelets aggregate at the injury site. They release factors that cause blood clotting, forming a fibronectin mesh. And this clot serves a dual purpose. It achieves hemostasis, so stops the blood loss, and it provides the initial provisional scaffold.
A track for the other cells to follow.
A track laid out for the subsequent cellular repair machinery to migrate across.
Phase 2 is inflammation. This is where the cleanup crew arrives.
Inflammation starts almost instantly. The initial cellular cleanup crew, the neutrophils, arrive first, within the first 3 days to clear out debris and bacteria. And following them, around day 3, the macrophages arrive.
And the macrophages are more than just cleanup.
Macrophages are the command center of the wound. They not only continue the cleaning process, but crucially, they release the necessary signaling chemicals, the growth factors.
And these growth factors are the signal to start the rebuilding phase.
Exactly. These signaling molecules, including those that stimulate new blood vessels and new cell proliferation, act as the command flare, dictating the tempo and the direction of the next phase. Proliferation.
Proliferation, phase 3, starts within days, and it has 2 essential tasks: closing the surface and rebuilding the support structure underneath.
First is reepithelialization. The keratinocytes at the wound edges, stimulated by those growth factors, change their shape, they break their anchors, and they migrate across that fibrin scaffold in a thin sheet to close the surface defect, thus restoring the barrier.
And at the same time, the underlying support structure is being rebuilt.
And that rebuilding involves granulation tissue. That's the second task. Granulation tissue forms as fibroblasts migrate in and begin synthesizing a new temporary extracellular matrix. At the same time, specialized growth factors stimulate angiogenesis, the budding and growth of new capillaries. This provisional, highly vascularized pink granular tissue is what fills the defect.
And finally, we enter the long-haul phase, remodeling.
Remodeling can extend from months to even years. It's all about refining and strengthening that new tissue. The fibroblasts differentiate into myofibroblasts, which possess contractile elements. And these cells actively pull the wound edges together, leading to the physical wound contraction that reduces the size of the final defect.
And the collagen itself changes structure to make the scar stronger over time.
Precisely. The initial granulation tissue is rich in a weaker, temporary type 3 collagen. During remodeling, this is gradually replaced by the much stronger and more durable type I collagen. But it's important to note, though, that even after years of remodeling, the final tensile strength of healed skin rarely exceeds 70 to 80% of the original undamaged skin strength.
That choreography is just astonishingly precise. So, shifting to clinical applications, we often hear about skin grafts and skin flaps. What is the essential practical difference between these two surgical techniques?
The difference hinges entirely on the integrity of the blood supply. A graft is a piece of skin epidermis and some dermis that is completely detached from its original blood supply and then moved to cover a defect.
So, how does that cut off piece of tissue even survive?
Grafts survive initially by a desperate race against time. For the first 48 to 72 hours, they live by plasmatic diffusion. Nutrients and oxygen just passively seep into the graft tissue from the recipient wound bed below.
And for long-term survival?
For long-term survival, it has to achieve revascularization. This means that new blood vessels from the recipient site must grow into the graft tissue and connect with its old vessels. It's a very tenuous process.
In contrast, a flap is moved with its blood supply already guaranteed.
Yes. A flap is tissue, which can be skin, muscle, or fascia, that is moved from a donor site to a recipient site while retaining its own inherent vascular supply. Because that blood flow is reliable from the moment of transfer, flaps can be much larger, much bulkier, and they're used when complex reconstruction is needed.
Like what?
Like covering exposed bone or joints, or repairing high-tension areas. Flaps offer much more reliable long-term tissue viability. So if we synthesize the key knowledge points from this deep dive, the human skin is clearly a unified, dynamic, and integrated organ system. It's simultaneously managing metabolism, temperature, immunity, and physical protection, and is coordinating all these complex actions at the microscopic cellular level.
And the incredible stability of the structure relies entirely on that precise coordination. You have the highly coordinated 28-day self-renewing keratinocyte barrier, which is cemented by the lipids and filaggrin from the granular layer,
Right.
sitting securely on top of that incredibly resilient dermal scaffolding made of collagen and elastin.
And understanding this structure fundamentally changes how we view common pathologies and even everyday phenomena. Why do we get persistent dry skin and eczema? Well, because of failure in the filaggrin machinery, a structural flaw in the granular layer that makes the skin leaky.
Right. And why do surgeons worry about tension lines?
Because the organization of those collagen fibers in the reticular dermis dictates how skin will heal and how it will scar.
And why do we blush or turn pale? It's because of the autonomic control over that vascular plexus and the AV shunts in the dermis, redirecting massive volumes of blood for a rapid emotional or thermal response. The level of engineering is just profound.
Absolutely. But even with this impressive self-repairing capacity, there's an intense area of ongoing research that suggests the skin holds far more potential for perfect regeneration than we currently utilize, and it really speaks to the future of healing.
You are referring to the untapped stem cell reservoirs.
Precisely. We know the basal layer contains stem cells for that routine turnover, but the most potent, most protected niches for epidermal stem cells are located in the hair follicle bulge and within the sebaceous glands. These are the deep, protected reserves of highly potent cells just ready to be deployed.
So these reservoirs are essentially the ultimate protected keys to perfect, limitless repair and regeneration.
They are the source of continuous hair renewal, and they are crucially mobilized to re-epithelialize massive, severe wounds like major burns. The biological capacity for perfect regeneration is already there. The challenge is figuring out how to fully activate and direct these intrinsic biological mechanisms at will to address chronic conditions and aging damage.
And that leads perfectly to our final provocative thought for you to consider as you wrap up this deep dive. If the skin's stem cell reservoirs, particularly in those protected niches of the hair follicle bulge, hold the key to limitless and perfect tissue regeneration, the ability to rebuild skin without scarring, what is the next frontier in utilizing this intrinsic biological intelligence to address severe burns, chronic non-healing wounds, or even deep structural photoaging? The capacity is already built into your body. The work now is in learning how to unlock it.